This shows the human embryo at exactly day 14 of development. At this stage, it is two flat sheets of cells floating in a fluid void. These two sheets make up the bilaminar disc. The top layer is the epiblast, and the bottom is the hypoblast. But you cannot physically construct a complex three-dimensional organism with internal organs out of a two-dimensional plane. To progress past the second week of development, these cells have to undergo a radical, complex spatial reorganization. The embryo must break its own symmetry, migrating and folding to transition from a flat surface into a true three-dimensional architecture. The event that solves this biomechanical problem is called gastrulation. Looking at this timeline, we see the first 2 weeks cover implantation and the bilaminar disc. Gastrulation initiates right at the start of week 3, specifically around days 15 to 16. Through a sequence of strict cellular migrations, the bilaminar germ disc is converted into a trilaminar germ disc. Every single tissue and organ in the human body will eventually develop from these 3 resulting layers. Gastrulation is the exact moment the anatomical blueprint of a human being is drafted. This top-down dorsal view of the epiblast at day 15 shows the trigger for gastrulation, a thickened linear band at one end called the primitive streak. As it forms, a central groove and an elevated circular node appear. That groove is the primitive groove. The circular apex is the primitive node, or Henson's node, containing a small central depression called the primitive pit. Before the structure appeared, the embryonic disc had no defined orientation. Now, with the streak in place, we have distinct perpendicular axes. The location of the streak physically dictates the caudal, or tail end of the embryo, automatically defining the cranial head end and the sides. The physical appearance of the primitive streak boots up the spatial coordinate system for the rest of human development. A transverse cross-section through the primitive groove reveals internal cellular movement. Epiblast cells migrate inward, detach, and slip underneath, a process called ingression. The first wave fully displaces the lower hypoblast layer, creating the innermost germ layer, the definitive endoderm. Meanwhile, a second wave dives through the groove, spreading laterally between the layers to become the intraembryonic mesoderm. Eventually, migration halts. The remaining surface epiblast cells solidify into their final position. This remaining surface layer is the ectoderm. Through the strict choreography of ingression, the embryo has physically built the 3-layered trilaminar disc. This structural rule of 3 stacked layers has 2 vital geographical exceptions. Looking at the full disc, there are 2 distinct circular zones where the middle mesoderm layer is completely absent. The cranial exception at the top is the prochordal plate, and the caudal exception at the bottom is the cloacal membrane. In these specific zones, the top ectoderm and bottom endoderm are fused directly together, with no mesoderm padding between them. This direct fusion creates a structurally weaker membrane by design. Later, these areas will rupture to form the openings of the mouth and anus. While gastrulation is actively occurring, we can observe a secondary migration if we look at a sagittal or side profile slice. Cells from the primitive node move straight forward beneath the surface. Starting around day 16, these cells form a solid, rod-like structure called the notochord. The notochord is a transient structure, acting as a master organizer. First, it defines the rigid central axis of the embryo. Then, it emits chemical signals upward into the overlying ectoderm layer. In a process called induction, the notochord signals the overlying ectoderm to physically thicken into the neural plate, initiating the creation of the nervous system. It also signals the surrounding mesoderm to begin organizing into the future vertebral column. The most vital permanent systems of the human body are architected by a temporary rod that disappears once its job is complete. This typographic layout maps the final fate of those 3 germ layers using a strict topological mnemonic: ectoderm covers, endoderm lines, mesoderm fills. Starting on the left, we have the rule ectoderm covers. This outermost layer gives rise to the central and peripheral nervous systems, as well as surface structures like the epidermis, hair, nails, and tooth enamel. Moving to the right column, we have the rule endoderm lines. The innermost layer forms the internal linings of the gastrointestinal tract and the respiratory tract epithelium, along with the liver and the pancreas. Finally, the middle column follows the rule. Mesoderm fills. This middle layer builds the structural and connective tissues, all muscles, bones, cartilage, kidneys, the heart, and blood vessels. A single cell's final destination in the adult body is strictly dictated by its physical spatial position at the end of week 3. Because gastrulation is an incredibly fragile biomechanical event, structural failures at this stage cascade into severe clinical pathologies. For example, the primitive streak must normally regress and disappear completely by the end of the 4th week. If the streak fails to regress, leftover pluripotent cells can grow into a massive sacrococcygeal teratoma. This is a tumor containing random tissues derived from all 3 germ layers. At the primitive node, a defect in left-right signaling can cause situs inversus, resulting in a literal mirror image reversal of internal organ placement. Further along, a failure in notochord signaling during induction leads to neural tube defects, such as spina bifida or anencephaly. The only normal clinical remnant of the notochord in adults is the nucleus pulposus at the center of your spinal discs. If other remnants persist at the base of the skull, they can form a rare tumor called a chordoma. By day 19, the success or failure of symmetry breaking, ingression, and layer formation has permanently sealed the architectural fate of the human life.